Hi, this is Mr. McLean, and I am back for our last section of review on the first unit in AP Bio, and that's going to be Chapter 5, Part 2, dealing mostly with the proteins. When we jump into Section 4 of Chapter 5, we're going to find out that proteins are really a critical concept all year long. And, in class, we talked about some of the different functionality that proteins serve. And if you review these few slides here, you're going to see that proteins, most of our enzymes are protein based. When we look at our immune system, whether we look at storage molecules, transport proteins, communication through hormones, receptor surface proteins, muscles, we usually think of, actin and myosin and motor proteins, structural things such as our collagen, things that make our connective tissue. So really, there's a significant number of structures and functions that are really, really dependent upon proteins. So the protein construction has to be right. And what we're going to see in this chapter is ultimately how are proteins assembled and what are the different levels of proteins that exist. So I think what we need to do is we need to start with what is a protein. And a protein is something that's going to be made up of polypeptides. So from your earlier bio days, you probably learned that you link up amino acids and you get a protein. And really what you're getting from the linkage of amino acids, you're going to get a polypeptide. And then as you go from there and you combine it and you take on your more complex bonding and three-dimensional shaping, then you get a protein. So it really only becomes biologically functional after you go through many of those different changes. So at the heart of our protein, you have the amino acid sequence. And this is what's determined by our DNA. So if you have a mutation in any way, shape, or form, that's going to alter whether or not you get the correct amino acid sequence. And that's really level one, which we will be talking about. This picture right here is going to show us what a amino acid is going to look like. And the thing that stands out as the point that's different from every amino acid to the next is the R group. Everything else below that is universal. So you're always going to have an alpha carbon in the middle. You're always going to have a carboxyl group. And remember, the carboxyl is donating hydrogen. So that takes on more acidic properties, but the opposite side is going to be more of a base. The structure, the backbone structure of amino acids will be universal. It's the R group that gives it properties that are different. So when we review this chart, which is found in our book, and we go through here, there are 20 different unique amino acids that are all different only because of that R side chain. So within that grouping, you find some that are going to give it hydrophobic characteristics. And it's not necessary for you to memorize which ones are hydrophobic or which ones are hydrophilic, but you just kind of look at what are the properties of some of those functional groups. And, for example, as we look up here, we see these methyls over and over again. And we learned that methyl was a functional group that was definitely going to be nonpolar. So as a result, we get those hydrophobic characteristics. We get down here, and we're seeing things like alcohol groups, all right? And we know that alcohol or carbonyls are going to give it more polar characteristics. When we go down at this next section, we take a look at it. We find that some of them give properties where they end up acting like an acid. So you're going to have these side chains that are, in some cases, going to be negatively charged. And we have a couple examples there. In fact, we refer to them as an acid. And then the last group, we have things that are going to actually remove hydrogen from a solution as they ionize, which you can see right here. You're seeing the formation of those positively charged side chains. So they're going to be more basic. So the idea is the R side grouping is really what's going to give it uniqueness, but the main structure is going to be the same pretty much in all the scenarios. So the bonding, much like what we saw with the carbohydrates and what we observed when we were talking about the the formation of the more complex lipids, is that you're going to have a specific type of bond that's going to show up in there. And the bond that we're looking at right here between different amino acids is going to be your peptide bond. So let's look at a formation of a peptide bond. And in this case right here, we've got a couple amino acids, and we're going to combine our third amino acid to make it a growing polypeptide. And once again, we see our very, very common type of chemical reaction. So anytime you're going to pull that water out of there, you got a condensation reaction. So this bond is what we would classify as a peptide bond. So as I mentioned before, the idea of being able to put those specific amino acids in order is critical, and it's first laid out by the DNA. But that really doesn't take us all the way to the end of the story. We need to make it a functional protein. We need to organize it precisely. We need to fold it and twist it and coil it. I would suggest highly that you review page 80 in your textbook, because it has a chart that reviews the 4 levels of structure that we're going to go over. So when we look at it and we move our way through this, what we're going to find is ultimately it is that sequence that's going to play a really key role in determining what the 3D structure is at. So basically, if you have level 1 wrong, if you have primary structure wrong, you have a mutation in there, not only is it going to be the wrong amino acid sequence, it's going to have secondary, tertiary, and potentially quaternary effects as well. So we have some significant examples where a single nucleotide change, which ultimately gives us the wrong amino acid in a sequence, is going to have really, really dramatic impacts. So if you look at it here, our 4 examples that we have: primary structure, it's all about having the unique right order, the right sequence. So your book talks about an example of a particular blood-based protein that has 127 different amino acids in the order, and they're in a very specific order to be correct. There's 20 different amino acids, so if we were just going random chance, you would have 20 to the 127th power different options of what you could get if it was totally random. So clearly, our DNA is very precise, and it's critical that we get a very, very accurate ordering of those amino acids to get the primary sequence. Secondary structure is we're going to start to form folds in different types of coils. So you're going to hear me talk about alpha helixes and beta pleated sheets. And the big thing that they're going to do is they're going to impact some of that 3D structure. And those are changes that happen because of hydrogen bonds that occur between the main chain of the polypeptide. We'll look at that. Tertiary, those R chains that we talked about a few minutes ago, those R side chains start to interact in different ways, like van der Waal interactions, and that really gives it more of that 3D shape. And then the final level, quaternary, is when you have multiple components that come together to give you something complex. All right, so if we look right here, this is primary structure. So this is that molecule or that complex protein polypeptide building block where all of these amino acids have to be right. Okay, so for this particular protein, that's the sequence. And you have one end that's going to start with an amino end, and one end that'll finish with a carboxyl end. So the terminal end is going to be the carboxyl end. So that's determined by our genetic information. Now, the next level is secondary structure, which we will see 2 key types of structures appearing. You get alpha helixes which obviously are going to give you kind of that helical configuration. So they call them the alpha helixes. And then the sheets are called beta pleated sheets. And both of them are going to occur because of hydrogen bonding. So you want to make sure you know that secondary construction is made up of hydrogen bonds that create alpha helixes, beta pleated sheets. So as we go through here, we mentioned that make sure you understand it's the backbone, the polypeptide backbone, that's really where you're seeing that at. And as we mentioned, a lot of times, depending on the complexity of the protein, we might have multiple sheets set up, or we might have multiple helixes. All right, so when we go to the next level, we're looking at tertiary structure. And when you look at tertiary structure, we're concerned with those R groups. And the idea with those R groups is sometimes they start to bond differently depending upon what kind of chemical properties they have. So sometimes we have what are referred to as hydrophobic interactions, where you get a cluster of different side chains that are going to kind of work together, and once they get real close, they have these van der Waal interactions that kind of pull them together. We also get covalent bonding, referred to as disulfide bridges, which kind of help reinforce that overall structure. So the idea of the tertiary structure is that you've initially set out primary structure, which is you have the right sequence. Then you have the sheets and the coils that kind of form in the alpha and the beta level. Those are hydrogen bonds between the backbones. And then you take those side groups and you start to let them bond with each other. And when it does that, you're starting to see that tertiary structure is really all about getting a 3D structure. So we're getting something that's going to be more complex, and it's going to be more capable of getting us to that level where we have a functional protein. So these are examples of the disulfide bridges that would form. We talked about ionic bonding earlier in the course. You got some ionic bonds that form here. These are those van der Waal interactions between the hydrophobic interiors. So a lot of times they get clustered inside because there's no water in there. And you even have some hydrogen bonding. So you have a variety of bond types. The big thing to remember is you're dealing with, the R chains. All right. To make a real protein, you got to get to level 4, which is sometimes you're going to have multiple chains and multiple structures that are going to come together. If you look at this picture of collagen, you have multiple fibrous levels that are going to come together. If you look at this picture right here, you're going to have 4 polypeptides that are going to come together. If you look at hemoglobin, you've got multiple subunits that come together. So the point is, you basically go through level 1, 2, 3 to get, a certain level of structure, and then you put multiple levels of those tertiary structures together to get our full-fledged quaternary structure. So quaternary is anytime you have 2 or more polypeptide chains and they make this big macromolecule. So our 2 examples that we've looked at, we got collagen. There's literally going to be 3 different types of polypeptides kind of making up a rope structure. And you have hemoglobin, which transports oxygen in our blood. And they're going to have alpha and beta chains that are going to kind of work together. All right. So if you look at a disease such as sickle cell, really this is a dramatic change in function because of a single amino acid change, which means that we end up with the wrong type of primary structure. So here we are. That change created a different position, number 6. We have that valine substitution. And what does it do is it completely changes the orientation of where the hydrophobic region's at, which gives it a different structure overall because of the way the bonding takes place. So if you look at a cell that is a sickle cell red blood cell compared with a normal red blood cell, you're going to see a dramatic change in the structure, which correlates with a lot of those different problems that people suffer when they have sickle cell. A couple other terms to make sure you know. There are environmental factors such as pH, salt, temperature. If you lose the structure, it's called denaturing it. And then the other thing that we have as we wrap this up is improper folding.